everyone, our topic is nucleic acids. So,
Okay, so first we have to talk about genes. So what are genes? They are a discrete unit of inheritance consisting of DNA that programs the amino acid sequences of polypeptides. So DNA, it's a type of nucleic acid, obviously. It provides directions for its own replication, and it's in a double helix shape. And it has adenine, thymine, cytosine, and guanine monomers, which are the nucleotides. And then RNA is ribonucleic acid. It's DNA transcribes into RNA, and then RNA translates its amino acid sequence of polypeptides into something that the proteins can use. And then it's usually one polynucleotide strand, and it also has adenine, cytosine, and guanine, but it also has uracil instead of that. And the nucleotides are monomers of DNA and RNA, and it consists of a 5-carbon sugar linked to a phosphate group and a nitrogenous base. And then the double helix shape that I was talking about earlier with DNA, it's 2 polynucleotides wrapped around each other, and then it has a sugar phosphate backbone while base is paired in the center.
So we can easily describe the relationships between RNA, DNA, genes, and proteins through 2 processes called transcription and translation. So what transcription is a certain part of a gene sequence is taken and replicated into a sequence called an RNA, and when it's completed, it's called an mRNA, which is a messenger RNA, which travels out of the nucleus where it belongs and goes to the cytoplasm where it codes proteins. So translation is when the mRNA goes to the cytoplasm and, with the help of ribosomes, codes a specific type of amino acid. So let's talk about nucleotides. So the structure of nucleotides all look similar, but they each have 3 different parts, which are the sugar, the phosphate group, and the nitrogenous base. So the sugar comes in 2 forms, either deoxyribose or ribose, deoxyribose being in DNA and ribose being in RNA. The only difference between these 2 sugars is that hydroxide is, one of the hydroxides on the bottom becomes a hydrogen on the DNA. So the phosphate groups are all the same on all of the nucleotides. They're coming off of the HOCH2 side of the sugar. They have 2 negatively charged oxygen ions, and this and the sugar are both hydrophilic, so together they make the backbone of the DNA. The nitrogenous base is it's what it makes each nucleotide unique from each other. So as we know, there are 8 nucleotides, but each one of the nitrogenous bases are a little different from each other. There are 2 different types, which are the purines and the pyrimidines. They make up they allow for the different bonds which happen in the DNA structure. These nitrogenous bases are bonded to the other side of the sugar, and these are hydrophobic.
so this would be ribose and guanine, deoxyribose and guanine.
And then this is ribose and adenine and deoxyribose and adenine.
This is deoxyribose and thymine. This is ribose and uracil.
And this is ribose with cytosine and deoxyribose cytosine.
So Tiger mentioned pyrimidine and purine. So these are 2 types of nitrogenous bases. So a pyrimidine nitrogenous base would have just 1 ring. So here you see this guanine it seems to have 2 rings, right? But this one, sorry, where's cytosine? This one only has 1.
Cytosine.
Right? This one only has 1. So cytosine, thymine, and uracil only have 1 ring, meaning they have 1 pyrimidine ring. This is what a pyrimidine ring looks like. But purines are different. Purines include adenine and guanine. They have 2 rings. They have 1 pyrimidine ring fused to an imidazole ring, right? So that's how they differ. So when they hydrogen bond, a purine always hydrogen pairs with a pyrimidine, which is why you see A with T, C with G.
So as Aditi just mentioned, you have those specific bonds in between the A and the T and the C and the G. So when this happens, you get something that looks like this. What you would see is this. This is flat, obviously not DNA. So what happens is when they bond, when the hydrogen bonds form, they get skewed, but instead of just twisting a little bit, it has to completely turn so that all the atoms have a place to be without running into each other, and that creates the double helix, which I just broke. And here we see that with these 2, there's only the 2 hydrogen bonds That's in between the A and the T, and between the C and the G, you have all the three.
So I'll be showing a dehydration reaction between the adenine nucleotide and the cytosine nucleotide. So the first thing that you're going to do is you're going to take off one of the hydrogens on the sugar, and you're going to take off one of the, this is an ionized phosphate group, but 2-, but this would have an oxygen and a hydrogen, and you would take the oxygen and the hydrogen off, so you would have water, and then this would bond. So this is a dehydration reaction. So now you have two nucleotides that are bonded to each other, right? You see the sugars, this is the backbone, and then you see the phosphates that alternate between the sugars, and that's the phosphates in the sugar backbone, so the phosphate-sugar backbone. And you see the two nitrogenous bases protruding, and these would be towards the center, and on the other strand, they would be towards the center, and they would pair. So for